Okay, we're going to start our lecture on the respiratory system. This will be a nice review because we've already gone through at least a majority of the anatomy in lab. So review the lab lecture. You can also look at the posted cadaver videos of the respiratory system, as well as looking at the visual body 3D animations to kind of remind yourselves of these respiratory structures. All right, so we've already talked a little bit about the functions of the respiratory system, but the functions of the respiratory system, number one, is going to be to exchange respiratory gases, oxygen and carbon dioxide. These are really important to facilitate aerobic cellular respiration, which occurs within all of our metabolically active tissue cells. Our metabolism or rate of energy utilization is very high, and so we need to make sure that our cells have adequate amount of oxygen to facilitate the most efficient conversion of nutrient to usable energy, which is ATP. Oxygen is required for this process and needs to be not only brought into our body, but distributed to all of our metabolically active tissue cells. And so the respiratory system's job is to bring oxygen in to our bodies from the outside air. That oxygen is going to come in at about 21% oxygen in the ambient air. As that oxygen goes in, it will diffuse in across our lung epithelial cells and into blood, and then circulate via the blood to our body tissue cells. When the tissue cells use that oxygen as part of cell respiration, that will facilitate the breakdown of nutrient energy, which are essentially as strings of carbon. And when those strings of carbon coming from our carbohydrates and our proteins and our fats, primarily carbohydrates and fats, are broken down, those strings of carbon are going to be released as CO2, which will then travel via the blood back to the lungs and then exhaled out into ambient air, which only has about 0.35% carbon dioxide. I know that most of us think of the respiratory system as bringing in oxygen, but the removal of carbon dioxide is a very important component of this process because carbon dioxide is extremely soluble. In fact, about 70% of the carbon dioxide that exists within the fluid of our bodies is in the form of bicarbonate, not actually in the form of carbon dioxide. And that's because carbon dioxide combines really easily with water to form carbonic acid that then gets broken down to bicarbonate and hydrogen ions. Because of that, the hydrogen ions will contribute to the overall acidity of the blood. And the more carbon dioxide we have, the more hydrogen ions that will start to accumulate and the higher the acidity of the blood. So breathing, in fact, is primarily regulated, meaning your breathing rate, by pH receptors. They'll monitor how acidic our blood is and therefore how much carbon dioxide we're producing by our metabolically active cells, and then adjust the breathing rate to make sure that we couple our energy utilization with delivery of oxygen. All right. In order to make sure we get a very efficient delivery of oxygen, we have to make sure that the lungs have an effective diffusion surface. The diffusion surface has to be a very large surface area, which on average is approximately 1,000 square feet of diffusion membrane. And it also needs to be very thin. They estimate that the distance that a single oxygen molecule or carbon dioxide molecule would have to travel across, it's 2 cell layers thick with a little bit of connective tissue in between. That's about 1/10 of a micrometer to about 1.5 micrometers. So very, very thin distance that we're going to be diffusing across. There's no specialized mechanism of sort of respiratory gas transport, no special proteins and things like that that help navigate them across the tissue membranes. But rather it's a concentration gradient. It's important that the respiratory gases are dissolved or solubilized, and then that there's a concentration gradient present. And so the gases simply diffuse, and the rate of that diffusion will depend on the thickness of the membrane and the concentration gradient and how soluble those gases are. Once we've diffused in across the lung epithelium, we also have to make sure that the lungs are perfused with a tremendous amount of blood, because we want to make sure that as blood comes into the lungs, it's about 70% oxygenated. That's actually venous blood or used blood. It needs to go from about 75% oxygenation up to 99% oxygenation to be effective for cellular respiration. And so we're going to see that diffusion distance and diffusion surface for effective exchange needs to be very, very thin and very, very large. So next thing is when we breathe air in, the air we breathe in is not clean. It has a lot of particulates in it and potential pathogens. So the air needs to be cleaned on its way in, because the reality is we're going to be 2 cells away from circulating blood, which will then circulate to within a millimeter of every metabolically active cell. So a pathogen is going to have access to pretty much anywhere in our body if it can diffuse across and into blood. So we need to make sure that the air we breathe in the process of carrying it into our lungs is going to be clean. We also need to make sure it's warm. Warm air is able to hold more water, and we need to make sure that the air we breathe in has a lot of water. For 2 reasons. One is we want to make sure that as we ventilate or bring air back and forth across our respiratory surfaces, we don't want that ventilation to dry out the very thin cells. And we also want to make sure that we can dissolve the respiratory gases. In order for oxygen to diffuse in our body, it first needs to dissolve in a very thin layer of water that sits inside the little respiratory passages. And so we need to make sure that the air we breathe in, by the time it gets to the exchange surfaces, is humidified. Next, we're going to look at phonation. Simple because we're breathing air, that air will allow us to cause vibrations. And so we're going to couple respiration with phonation. Phonation is the production of sound. It begins by the vibrations of our vocal cords and then gets modified by things like our pharynx, our soft palate and uvula, our nasal sinuses, our tongue, our lips, our cheeks. All of these will change the sounds that we produce so that we can have the variety of arrays of sounds that allow us to articulate words. And then last but not least is regulate blood pressure, blood volume, and pH. This may seem far-fetched because we look at the respiratory system as being involved in exchange of gases, and now we're talking about blood pressure and blood volume and pH. It doesn't seem like they're connected. But the lungs are part of a very important system of blood vessel pressure and our kidneys' ability to retain water and salts. And we mentioned with CO2 can regulate blood acidity or pH. The cells within the lungs, and specifically the little tiny air sacs, are called pneumocytes or alveolar cells. They have an important surface molecule called an angiotensin converting enzyme. And that name, if you listen really carefully, I said angiotensin. Tensin refers to tension, and angio refers to blood vessels. So the angiotensin converting enzyme converts a molecule called angiotensin 1 into angiotensin 2. And angiotensin 2 has an ability to tense or cause constriction of blood vessels, therefore elevating blood pressure. And it also can travel to the kidney and cause the release of a hormone called aldosterone. And aldosterone is a salt retainer. And when we retain salt, we also retain water. So therefore, it's a blood volume elevator. And so an important function of the lungs will seem to be not connected to respiration at all, but that will be to regulate blood pressure and blood volume and pH. All right, so here's a quick overview. This is a video you've already seen, so if you don't want to watch it, just skip past it. Otherwise, there'll be a nice review.
The respiratory system moves air through the nose, pharynx, larynx, trachea, and bronchus to the alveoli, where the gas exchange between oxygen and carbon dioxide occurs. Nares are the openings to the nose. The nasal cavity is lined with cilia, mucous membranes, and blood capillaries. The air is filtered by cilia, moistened by mucous membranes, and warmed by the blood. Air moves into the pharynx or throat, the common passageway for food and air. Air continues on to the larynx. The epiglottis, a flap of tissue in front of the larynx, closes off the larynx when swallowed to prevent food from entering. The larynx, or voice box, contains the vocal folds. The trachea, or windpipe, connects the larynx to the bronchial tree. The cartilage rings of the trachea prevent the trachea from collapsing. Lungs are spongy tissue with alveoli and blood capillaries. Breathing occurs because of the expansion and contraction of the lungs. The bronchi carrying the air subdivide into smaller branches called bronchioles. At the end of each bronchiole are the alveolar sacs. The alveolar sacs are surrounded by blood capillaries and contain millions of single-layer alveoli cells where the gas exchange takes place. Oxygenated air goes from the nose to the pharynx, larynx, trachea, bronchus, and alveoli. By the process of diffusion, oxygen in the air moves from the alveoli to the capillaries. Carbon dioxide moves from the capillaries to the alveoli and is exhaled. This process is called respiration.
Okay, so that's a nice overview of where we're going. So next we want to look at the upper respiratory system. Like I mentioned in lab, depending on where you cite or Google, upper respiratory and lower respiratory structures may have a slight difference in the structures they include. The upper respiratory system always is going to include the nasal cavity, paranasal sinuses, and pharynx. And the larynx is sometimes dropped down into the lower respiratory system and sometimes included in the upper. I personally like to include it in the upper. It doesn't really matter where you include it, but I like to think of the upper as being those structures outside of the thoracic cavity, and then the trachea and the branches of the bronchial tree and alveoli, that'd be part of the lower respiratory system. All right, so this is just a nice figure showing us again the entry point for air into our respiratory system. And there's two entry points. Obviously, the external nares or nostril goes into the nasal cavity, and then down into the nasopharynx, oropharynx, laryngopharynx, and then the larynx. And then, of course, you can also breathe air through your mouth. Larger gaps of air, especially if you're exercising, will go through your mouth. The downside with mouth breathing is obviously we don't have the same level of conditioning of air. Remember, the nasal cavity is going to have those wonderful coiled bones or nasal conchae. They're going to help begin the process of cleaning and humidifying and warming the air we breathe in. So if you breathe through your mouth, they're going to bypass that. Luckily, we do have a lot of cleaning and humidifying and warming that will continue along the transit through the conducting pathways. But realize that the nasal cavity is really designed to begin that process. All right, so nasal cavity is about air preparation, like I just mentioned. Again, we need to filter out inhaled particulates. So you have your terminal hairs here at the external nares. As we breathe in, I don't know about you, but I mentioned this before, I was extremely surprised at how large the nasal cavity is and how voluminous this chamber for beginning this process of cleaning is. The entire nasal cavity is going to be lined with a mucous membrane. It's going to go over those coils of bones. Remember, we have 3 coils, 2 from the ethmoid and 1 from just simply the inferior nasal concha itself. And these turbinates or conchae are going to swirl and sort of spin the air, so you get a lot of opportunities for particulates to get stuck in the mucus that's secreted by this mucous membrane. Mucus is a pretty sort of inexpensive substance to produce by the body, so it's a very easy way to get rid of a lot of particulate. We just simply produce mucus, gets stuck in the mucus, and then that mucus then travels down through our pharynx, and we essentially just simply swallow it. It does mean, though, that you can get infections within the nasal cavity. Those can migrate to the associated sinuses. In this mid-sagittal section, we can see the frontal sinus and the sphenoidal sinus here. Remember, laterally, we're going to have the very large maxillary sinuses, and you can get infections stuck in those areas, which can increase pressure as mucus production increases and cause sinusitis and headaches dealing with the infections in those sinuses. All right, so we're going to be able to inhale that air and we're going to start to filter it. We're also going to warm it as well as humidify it. We're going to find that, remember, right here is where we had that cribriform plate of the ethmoid bone. We're going to have little tiny olfactory nerves that come down into the roof of the nasal cavity. So one of the other reasons we breathe is so that we can have the opportunity to sense the particulates that have odor in what we're breathing. And so we get what we call olfactory sensations or smell. In the air preparation, again, I mentioned that we're going to bring air in. So we're going to filter through those external nares. We have those terminal hairs there as well as mucus. Humidifying the mucous membrane allows us to have a very wet surface. And again, I mentioned two really important reasons for that. One is to prevent the desiccation. That's a fancy word for drying out. When we ventilate, for example, if you wanted to dry something off, you often will blow across the surface. That's, passing air. When we breathe, that's called ventilation. That means that we're going to pass air quickly in and out of our respiratory spaces. And that movement of air can cause these very thin cells to dry out. And we don't want that damage to occur to the lung epithelium. And then I also mentioned that when we breathe air in, we need oxygen to diffuse. The oxygen in the air is a gas, and we need that gas dissolved in water so it can actually diffuse into the cytoplasm of the epithelial cell, into the cytoplasm of the endothelial cell, the blood vessel, and then into the plasma. So it needs to be dissolved, and so we have to have water, a thin layer of it, lining the inside surfaces of our diffusion membranes. And then warming is also really important because warm air is going to hold more water. This is really easily articulated or shown. If you take a nice hot shower, you'll recognize that heat rises. And as heat rises, you'll notice if you look at your mirror, if you have a large mirror in your bathroom, that the top of the mirror where it has the hottest air, you're going to see water condensing on the mirror. And that's because the higher the air is, the warmer it is, and the more moisture it's holding. And so when it hits a cold surface like your mirror, it's then going to cause the condensing of water. So you're going to see that there's more water in those warmer sections of air. So the more warm the air is, the more water it'll hold, which will allow us to have better dissolving of those respiratory gases. So warming comes in from the fact that we have a very highly vascularized membrane. So the warm blood from our body helps to warm that air. All right. In association with the nasal cavity, we have what we call paranasal sinuses. So remember, we have a lot of bones sitting around the nasal cavity. So we have our frontal bone, which is a cranial bone, the sphenoid bone, which is a cranial bone, and the ethmoid bone, which is a cranial bone. And then we have the maxillary bones, which are going to be facial bones. And the collection of these create these nice mucous membrane lined pockets within those bones that not only helps to reduce the weight of the skull, but creates these resonating chambers for phonation, as well as helps to keep warm, sort of humidified air. So helps that process of conditioning the air. So I have here sinus function, again, aid in phonation. If you ever had a sinus infection, you'll know your voice sounds a little different when those sinuses are full with mucus. So they create resonating chambers for our voice, also aid in humidification and reduce the skull weight. These are really nice drawings kind of showing you the frontal sinuses, the ethmoidal sinus, and the maxillary sinuses. And then in the mid-sagittal view, you can also see the sphenoid sinus. And so all of these are going to be lined with mucous membranes and allow for that phonation, humidification, and reduction of skull weight. All right, now we're going to move into the pharynx. Remember, the pharynx is this common passage that's divided into the nasopharynx, oropharynx, and laryngopharynx. The nasopharynx is the superior aspect of that pharynx above the soft palate. One structure that we didn't talk about yet are the pharyngeal tonsils. There's a tremendous amount of immune tissue associated with the entrance into our body. So we've talked about the palatine tonsils that sit between the platyglossal and palatopharyngeal arches, the lingual tonsil that sits at the root of the tongue, and now we have the pharyngeal tonsil that sits in the superior aspect of the nasopharynx. All of them are just aggregations of white blood cells that are encapsulated, so they have connective tissue around them. And these will help to fight off infections, recognize, I should say, and fight off infections when they come into the body. Often we'll refer to the pharyngeal tonsils as your adenoids. So if you hear anyone talk about their adenoids, they're talking about the pharyngeal tonsils. All right, associated also with the nasopharynx is a really important structure called the auditory tube or eustachian tube. We'll talk about this again when we deal with hearing, because the auditory tube, like the name implies, is going to be associated with the middle ear. It's really important in hearing that we have the ability to augment our sounds that are vibrations of air. And that augmentation or enlarging of the sound waves is done by the eardrum. The eardrum vibrates, and then as it vibrates, it then oscillates these little tiny ear bones that sit in the middle ear, and they will then transduce sound to our hearing structure called the cochlea. So what we're looking at in these drawings, here's the external auditory canal coming in. This portion right here is the middle ear. And then in purple here, that's the inner ear or cochlea, which is going to be where we actually sense sound. Connecting the nasopharynx to the middle ear, where these little tiny ear ossicles are, is the eustachian tube or the auditory tube. The eardrum is right here, which you kind of see on this figure, and right here on this one. And it's a very thin stretch of membrane. And we want to make sure that it vibrates equally. So we need to make sure that the pressure on either side of it is equal. And so that's really what the eustachian tube is all about. It's about equalizing pressure in the middle ear. As we go up in elevation or down in elevation, the atmospheric pressure changes. So if you've ever flown in an airplane, oftentimes you'll have pain in your ears. And that's because at higher elevations is lower air pressure. And so we're going to find that the eardrum will flex out because the inside pressure in your head is going to be greater than the lowering outside pressure as you go up in elevation. Oftentimes when this happens, you can say, well, I need to pop my ears. And what usually you do is you change the position of your mandible, like you rock your mandible back and forth or you chew. And that really stretches this eustachian tube as you're manipulating your pharynx. It kind of pops and stretches open. Oftentimes you'll hear it kind of crackle. You can even do that now if you want, just kind of adjust your mandible and you'll hear this crackling sound. And all you're doing is kind of opening this pathway by stretching and pulling on your pharynx. And what that does is it makes sure that the air pressure that's inside your skull is going to be the same as the pressure outside your ear. And so the eardrum then doesn't flex. If you've ever gone swimming and you dive down in the water, you'll recognize that you have that similar kind of pain in your ear. And that's because the lower you go and the more water pressure there is, the eardrum is going to flex in. And so that then causes pain. And so you can also kind of adjust it. It's a little bit hotter, obviously, because you're in water, you're not going to get that same equalization. But that's what we find the primary function of the eustachian tube is. Now, one of the biggest problems with the eustachian tube is that it's associated with your pharynx, which is this entry point for inhaled air and food, which means you can get bacteria and other kinds of pathogens entering in through that pharynx. And those bacterial infections can track through the eustachian tube and into the middle ear. And it's one of the ways you can get middle ear infections. In children, their skull is much rounder than when we're an adult. Our skull tends to narrow. And so the position of the eustachian tube in a child is going to be much more horizontal and shorter. And so it's a shorter route of infection, or say for infection to travel. And when infections occur, it causes swelling on the inside aspect of the eardrum, and it starts to flex it out, and that creates a lot of pain associated with ear infections. Adults can still get ear infections, but it's a little less likely because the eustachian tube is a little bit longer and a little bit more vertical, so it's harder for infections to track upward that distance. Okay, so we've looked at the larynx in lab, and if you haven't seen the video, I highly recommend you watch the video I posted on the laryngeal cartilages. But the larynx, commonly referred to as your voice box, or your Adam's apple, especially in males, because the cartilage of the larynx tend to enlarge in response to the presence of testosterone. You have a laryngeal prominence, which you can see, this is a drawing of a male. So this laryngeal prominence, which is called the thyroid prominence, is this anterior protrusion that often we'll refer to as our Adam's apple. The important aspect of the larynx is that it is a entry point into our respiratory pathways. And so one of the laryngeal cartilages that we've already talked about, which is the epiglottis, will serve to protect that entrance. But it also houses our vocal structures. So it will protect the entrance into the respiratory pathways and then house the structures involved in phonation. Location is right around C3 to C6. So you're in your cervical spine, and then sitting right above the larynx at right around C2, C3 is going to be your hyoid bone. All right, so here we can see a nice view of that hyoid bone. So here's the hyoid bone sitting right above the larynx. It's connected with this really thick connective tissue called the thyrohyoid membrane along the side here, and it gets really thick in the front, and they call that the thyrohyoid ligament. It will help to attach the whole larynx structure and the trachea that sits below it to the hyoid bone. The hyoid bone, remember, is the only bone in the body that doesn't articulate with any other bone, but it is attached with a lot of muscle. And those muscles will function to raise the hyoid bone. And because the hyoid bone is connected to the larynx, it pulls the larynx upward during swallowing. All right, so again, protect the entrance into the respiratory structures. You can see the little epiglottis, kind of looks like a tongue up here, but there's your elastic cartilage epiglottis kind of sticking up above the hyoid bone there. And then although we can't see them, our vocal structures are going to sit inside the larynx there. All right, so I have here the protected entrance. So remember, the common term for the entrance into the respiratory pathways is the glottis. The structure that protects that entry is the epiglottis. Remember, epi means above. It's sort of a horseshoe-shaped elastic cartilage. And when we swallow, as the larynx gets pulled upward, it just simply folds backwards or posteriorly over the opening to the larynx or the glottis. This is a laryngoscope, and we'll show you a couple of videos. I showed them in lab, so again, there'll be repeats, but it's nice to see them again. But a laryngoscope is put into the oral cavity, and it has a little lighting camera, so we can look down into the laryngopharynx down here. And this is the view we're getting. So this right here is the epiglottis. This is anterior. This is posterior here. So if we were to push further back, this would be where the esophagus is beginning. Sitting in front of the esophagus is going to be the respiratory structures or the larynx. And these are the vocal folds, this sort of wider mucous membrane covered fold here. So these are the true vocal folds. And then these are the vestibular folds. And if we open up and look through the glottis, we can already see the little cartilaginous rings of the trachea sitting anteriorly there. All right, so this is a quick video on epiglottis. Again, you've seen it, so if you don't want to watch it again, just fast forward.
Inhaled air and ingested nutrients both pass through the oropharynx behind the oral cavity. A cartilaginous structure called the epiglottis directs food and fluid away from the trachea and into the esophagus, preventing inhalation of this material. The stem of the epiglottis attaches to the hyoid bone and the anterior rim of the thyroid cartilage. The superior portion of the epiglottis moves freely and can swing up or down like a trap door. With each swallow, the larynx rises and the epiglottis folds down over the laryngeal opening, closing off the airway. If particles make their way into the trachea, the cough reflex pushes air forcefully up through the larynx, forcing the particles back up and out.
Okay, so nice animation, but just one note. Remember that when you swallow, the uvula here is also going to be critical in making sure that food doesn't go into respiratory pathways. So remember, the uvula is going to contract and close off the nasopharynx so we don't get food going up into our nasopharynx. The video didn't show any movement there of the uvula, but we know that's going to happen during swallowing as well. All right, so we spent a lot of time in lab looking at the larynx, so you should be able to name all these amazing laryngeal cartilages and structures. So remember, of course, the hyoid bone, which is not the larynx, but is very important in the association with the laryngeal position. We have the very large anterior thyroid cartilage, the largest of all of the laryngeal cartilages. It's a very broad kind of U-shaped cartilage that sits anteriorly with its laryngeal or thyroid prominence, and then has this nice big sort of lateral sides, and in the back has this cornu or horn that helps to attach it to the hyoid bone. Underneath the thyroid cartilage is the cricoid cartilage. Notably, it's the only ring of cartilage that is complete in the entire respiratory system, meaning that's an entire ring of cartilage. So even though we talk about the tracheal rings, they're not complete rings of cartilage. They're going to have gaps posteriorly. And then we can see the little epiglottis sitting up here. In the parasagittal section here, we can see, again, the cricoid cartilage, which is going to be narrow in the front and have a really broad base in the back. The cricoid cartilage is going to have sitting on top of it the arytenoid cartilages, which are kind of pyramidal in shape. And then on the top of the apices of the arytenoid cartilage up here will be the little tiny corniculate cartilages. And in the superior fold of the glottis here, this little lump here is going to be the cuneiform cartilages, which just kind of makes this edge a little bit stiffer. Okay, so total of 9 laryngeal cartilages. We have 3 unpaired cartilages. Again, the epiglottis, thyroid, and cricoid. And then we have 3 paired cartilages, where we're going to look at the arytenoid, corniculate, and cuneiform cartilages. All right, and then just a nice picture. This is out of your textbook again, showing the epiglottis folding back here posteriorly during the swallowing or deglutition of this bolus of food. We can see the bolus would be forced posteriorly into the esophagus from the oral and laryngopharynx. And nicely here we can see the soft palate and uvula moving posteriorly and superiorly to make sure that we close off that nasopharynx during swallowing. All right, so for phonation, we've talked a lot about this in lab, but the idea is when we vibrate structure, that produces sound waves, and those sound waves or vibrations can then be manipulated as they travel from the sound-producing structures, which are called our true vocal folds or cords, out of our body. So again, that manipulation is going to occur with your lips and your tongue, your pharynx, your soft palate, uvula, the resting chambers of your paranasal sinuses will all affect what those sounds will be produced, or which sounds will be produced. All right, so the composition of the vocal folds are really a very thin mucous membrane will cover them, and they're going to be made up of an elastic ligament and some smooth muscle. And then we have the pitch, skeletal muscle. And then we have the pitch that gets altered by the tension of a ligament. And what we're going to do is we're going to use laryngeal muscles. Again, these are going to be skeletal muscles that we use to change the pitch of the sounds that we produce by tensing or relaxing the tension on those vocal folds. All right, so the other component involved in sound production, which actually serves really as a protectant, are going to be the superior located vestibular folds, sometimes called ventricular folds. But vestibular folds, you can see them labeled here, are these much fleshier superior folds that sit right above the thinner vibrating folds. These fleshy vestibular folds are really designed to protect the vocal folds that are going to be producing sounds. So as we see here, they're sometimes called ventricular because there's a gap between the true vocal fold and the false vocal fold. So you can kind of put your finger, if you were inside here, that's called the ventricle. And so sometimes you'll see this labeled the ventricular folds. All right, and again, this is a view looking if you had a laryngoscope inside someone's larynx. This is the anterior because this is actually the root of the tongue. Here's your epiglottis, and then this is posterior. This is the back end of the larynx. So these would be the corniculate cartilages sitting on top of the apices of the arytenoid cartilages. So this would be the position of the vocal folds when you're taking a nice big breath, right, moving air. And this would be the position of the vocal folds when you're actually producing sound. So you need the cords to be in the pathway of air in order to vibrate. So this would be sound production, and this would be during breathing. All right, so this is a little quick video on phonation.
The larynx is a critically important structure for both life and for speech. It is buried deep in the tissues of the neck. The larynx is made up of a series of cartilages placed atop the trachea. The cricoid cartilage sits immediately on the trachea, and the thyroid cartilage articulates with the cricoid. The tiny arytenoid cartilages sit on the posterior cricoid cartilage. The vocal ligament of the vocal folds runs from the arytenoid to the thyroid cartilage. The lateral cricoarytenoid muscle closes or adducts the vocal folds, rocking them toward the midline. Phonation for speech is initiated by simultaneously starting airflow from the lungs and trachea and bringing the vocal folds closer together. When the folds move closer together, the air pressure between them drops, and the vocal folds are sucked into the airstream and are pulled closed. The air pressure beneath the folds blows them open, but they are pulled back together by elasticity and the negative pressure of the Bernoulli effect. This cycle is repeated over and over until the vocal folds are abducted or removed from midline. When you look at the vibrating vocal folds in cross-section from behind, notice that they open from the bottom to the top, and that they close from the bottom to the top. This is a critical contributor to the complex acoustical phenomenon of voice.
Okay, so this is going to be that endoscopic view looking in. And so what we're going to do is you're going to look inside through a laryngoscope, and then you're going to see the epiglottis, and then you're going to see the opening, which is the glottis, the true and false vocal folds, and then you'll be able to even see the little mounds of where the corniculate and cuneiform cartilages are. So we'll play this.
Nasal drainage. This is the hardest part. I know it's going into your throat. That's the only part that's a little painful. Relax. Relax with me. I know. It's over.
So you're looking down into the larynx below. So you're sitting in the throat, in the oral cavity. There you go. And so you can see the epiglottis in front.
You know, don't swallow.
So there's that epiglottis right there. So this, you can see these are the arytenoid cartilages right there.
Higher pitch.
Right there. These are corniculates, sorry. Oh, no, sorry, cuneiform. And these are the corniculates back there. False vocal folds and true vocal folds.
That phonation, inhalation. Good. Just keep.
And this back here, that's the esophagus. That would be the opening to the esophagus back there.
Start low and go high for me. How about one fluid note, low to high? Do anything you want. Anything you want on tape.
There's a bright golden haze on the meadow. There's a bright golden haze on the meadow. The corn is as high as an elephant's eye, and it looks like it's climbing clear up to the sky. Oh, what a beautiful morning. Oh, what a beautiful day. I've got a beautiful feeling. Everything's going my way.
Don't follow. One more time. E. Anything else?
All right, so we had a really great view of the larynx from a sort of a laryngoscope point of view. So we're looking down into the larynx and looking at those laryngeal cartilages. These are just really nice drawings where we're able to see in color the 9 different laryngeal cartilages. So remember, we have 3 unpaired cartilages: epiglottis, thyroid, and cricoid. So this is looking posteriorly. And then we have the paired cartilages, which are here the arytenoid and the corniculate. And then we can't see the cuneiform because they're in that mucal fold, so those usually get removed. Remember that the thyroid cartilage is really a U-shaped cartilage, so the larynx in the back is pretty open. In that video, you could see that big gap behind the cricoid where we're going to be passing into the esophagus, okay? All right, so laryngeal cartilages, this is a quick review. The largest of the laryngeal cartilages is the thyroid cartilage. It's the large anterolateral prominence that we palpate when we're feeling our larynx or our voice box. So here's a really nice kind of view of it being this big U-shaped cartilage. It kind of goes around. It creates a nice anterior protected sort of plate of cartilage for the vocal folds that will sit inside. The thyroid is going to function as an attachment point for the epiglottis, as well as the vocal structures. We're going to see have some attachment into that thyroid and cricoid structure. And we're going to see that the thyroid cartilage is going to sit superior to the cricoid cartilage, which sits right below it. All right, so functions are going to be to support the vocal structures, to protect the opening. Obviously, we need this very large anterior plate of cartilage. So, you know, if you are touching, you can kind of squeeze your larynx a little bit and adjust the sounds of your voice as you're flexing some of the vocal structures. And then again, it's connected to the hyoid bone. So it's really what we pull when we're swallowing to help move the larynx upward and fold that epiglottis posteriorly. All right, so this is a view here. So this is the thyroid cartilage right there, and we can see the connection here to the thyroid cartilage for the true and the false vocal folds, okay? All right, the cricoid cartilage, like I mentioned, is the only complete ring of cartilage in the entire respiratory system. It supports the vocal structures. So remember, sitting on top of it will be the arytenoid cartilages, which are going to be the primary cartilages in adjusting the pitch of sound and also moving the vocal structures actually into the pathway of passing air. Also helps to protect the glottis and attaches to the trachea. So the trachea is going to be the next segment of the conducting pathways of the respiratory system. And we're going to see that there's a cricotracheal ligament that connects the trachea to the undersurface of the cricoid. All right, so this is looking at the cricoid cartilage. You can see again, it's this complete ring. This is the anterior edge of it, small in the front and very tall in the back. Here we can see where the two arytenoid cartilages are going to sit atop. This is actually a synovial joint, even though it's between cartilages. As those cartilages move back and forth, we want a nice, similar to any bone that moves in a joint, we want to make sure those cartilages are going to move with a nice fluid movement with no abrasion. So we're going to actually have a synovial joint there. We're going to find that we have cricoarytenoid muscles that will help to move the arytenoid cartilages and navigate the positioning of the vocal folds. This is looking at the cricoid from the front. So we can see the cricoid cartilage here, and then the cricotracheal ligament holding the cricoid to the trachea. All right, next to the arytenoid cartilages, these are the first of our paired cartilages. They're really important in regulating the tension of the vocal folds and moving the vocal folds into the pathway of passing air to create sound and vibration. They're pulled by arytenoid muscles. One of the most important is the cricoarytenoid muscle, but we also have thyroarytenoid muscles that are going to help to affect the tension and positioning of the vocal folds. And in the back here, these are really nice views of the stripped down pieces of cartilage. They're pyramid in shape. They're pointed at the top, so these would be the apices, where we're going to see the next set of paired cartilages actually going to sit atop them. So these are the corniculate, because they look like little horns. Cornified, remember, means horned. So these are the corniculate cartilages that sit atop. And this is the vocal ligament that's attaching to the anterior edge of the arytenoid and the posterior middle or medial edge of the thyroid cartilage. All right, this is looking from a superior view down on top of the arytenoid cartilages here and here. You can see coming off this anterior edge of the arytenoid is the true vocal ligament attaching to the medial posterior edge of the thyroid cartilage. And then we can see the two little corniculates that sit right atop of the arytenoid. And so we're watching sound production. You're going to see those two corniculate cartilages in the back. And the more obvious structure, at least bump that I typically see, is the cuneiform. But in all of these stripped down versions where they pull the mucous membrane off, you don't see the cuneiforms. So in this view, I don't see those cuneiforms. And yet when the mucous membrane's there, they're very obvious. And that's because they're really sitting in that fold of the mucous membrane. So if we were looking for the cuneiforms, they're going to be right here in the mucous membrane. But when we pull the mucous membrane off, those little cartilages don't articulate with any other cartilage, and so they typically are not drawn. This is a really great view of that U-shaped thyroid cartilage. We see the ring of the cricoid cartilage, and then we see the two pyramidal arytenoid cartilages and the two smaller corniculate cartilages, again with the vocal fold or vocal ligaments right there. All right, and then again, the opening to the respiratory pathway, this is the glottis. All right, and then this is a mid-sagittal section, again, just showing you the true vocal ligament and then the superior false vocal ligament. Here we can see the arytenoid cartilage with the corniculates kind of right on top. And then we have the cricoid, which is really tall in the back, supporting the arytenoid cartilages, and then small in the front. And then we can see the cricotracheal ligament right here connecting the trachea, which is going to be just inferior to the larynx. And then last but not least, we can see here, here's your epiglottis that's going to be attached right there to the inside edge of the thyroid cartilage. All right, so the corniculate and cuneiform cartilages are two more of the paired cartilages. Again, the corniculate are going to sit right on top of the apices of the arytenoids, and then the cuneiform are sitting in that superior edge of the mucal fold. So in drawings where they only show the cartilages with no mucous membrane, you're not going to see the cuneiforms. But when we see the mucous membranes and mucal folds, they're very obvious. In dissection, though, because the mucous membrane doesn't have any tension in it, they're really hard to find. So in the video that I showed you, we're not even seeing the cuneiform. So I'll show you in that video the thyroid, the cricoid, the arytenoid, and the corniculates. Okay. All right, and then the epiglottis we spent a lot of time talking about. Again, that's one of the unpaired cartilages that functions to protect the opening or the glottis from when we're swallowing ingested substances. All right, and so these are pictures, and these big bumps here and here, those are the cuneiforms. So they seem super obvious when you have them fleshed or you have the mucous membrane. And then back here, these are going to be where the corniculates are, and they're super obvious in the drawings of the cartilages, but not quite as apparent when you're looking at the mucous membrane covered structures. So we have the cuneiforms. Well, where's it go? The cuneiforms are these guys right here and right here, and they kind of help tense the super edge of the mucous membrane. And then we have the arytenoids back here. So this is posterior. We have the true vocal fold, and then we have the false vocal fold. And then there's the epiglottis. Okay. All right, so this will be one more laryngoscope. This one I did show in lab. So again, if you don't want to see it, you can fast forward.
Here is a look at some normal young female vocal cords during a stroboscopic exam.
So again, these are the these are the cuneiforms right there. You can see the true vocal folds, false vocal folds. Here's your epiglottis. Back here, these are the corniculates. And if we were to pass back here, hopefully we'll get another view. That's the opening to the esophagus. So notice the opening here, that's the glottis. The arytenoids are pulled inward to bring the vocal folds in the path of sound. And that's the esophagus back there. All right, so again, just reviewing, so make sure you feel comfortable recognizing and either labeling or describing the position of these different cartilages. All right, so now we're going to move down to the lower respiratory system. So we're going to look at the trachea, the bronchi or bronchial tree, and then structures of the lungs. So remember that there's the upper respiratory system and then the lower respiratory system. The upper respiratory system is purely conducting air in. There's no exchange of gases that takes place in the upper respiratory system. So it's really all about cleaning, humidifying, and preparing air, as well as being involved in phonation or the changing of sound. The lower respiratory system is going to have both a conducting zone as well as a respiratory zone, meaning we're going to continue to condition air, and then we're also going to finally get exchange of gases, which is what we refer to as respiration. So we'll have conducting zones and respiratory zones. So the trachea is purely conducting. It's merely a tube that passes air from our pharynx down into the substance of our lungs. The trachea is about 4.5 inches long in the adult and about 1 inch wide. It's always going to be open, just like the trachea, or sorry, just like the larynx is always open, and your nasal cavity is always open. We want to make sure that air moves in with the least amount of resistance. And so we need to make sure that those pathways are always open and that they don't have to be opened in order for air to be moved in and out of our bodies. So the trachea is held open with C-shaped cartilaginous rings that essentially sits anterior to the esophagus and runs from just below the larynx, so around T4, around C4 all the way down to about T4, C6 down to T4 or 5. So when we're looking at positioning of the trachea, again, let me say it again, the trachea runs from about C6, C7 all the way down to about T4, T5. And if we think about externally, right below where the sternal angle is, so where your sternum's manubrium and body sits, that's where we're going to see the trachea bifurcate and start to become what we call the bronchial tree. All right, so that's what this is looking at, but that was hard to say. So again, the trachea is going to sit anterior. Again, it's about 4.5 inches long, running from about C6, C7 all the way to about T4, T5. We're going to see that if we had the sternum placed here, this would be right where the manubrium is, and then you'd have the sternal angle here, and this would be where the body of the sternum is. So this is the bifurcation. So this would be the end of the trachea and the beginning of what we call the bronchi. This last little piece of cartilage, we mentioned that in lab, that's called the carina. And once we pass the carina, which is sort of this V-shaped cartilage, we then enter into the bronchi. All right, so it's a flexible tube. You can, move it in your throat. So we're able to adjust the position of our neck and head. It is held open with those C-shaped cartilaginous rings. There's usually between 15 and 20 of them down that 4.5 or so inch distance. They're not complete rings. They're going to be open in the back. And remember that the back edge of the trachea, or the posterior side of the trachea, is actually going to be completed by a muscle called the trachealis muscle. So this is smooth muscle that sits across this posterior edge of the trachea. So this is trachealis. And then right behind the trachea, again, it's going to be our esophagus. So here's the esophagus. That's a really nice drawing, so you can kind of go through the esophagus again. I remember your muscularis externa with their intercircular and adelongitudinal muscles. This is all submucosa. There's that muscularis mucosa, and then there's the mucous membrane. Remember, the esophagus is stratified squamous. That's why it has a really thick epithelium here. So there's your esophagus. It's going to sit right behind the trachea, which sits right here. The trachea is going to have this wonderful hyaline cartilage C-shaped ring. And then we have a mucous membrane here. You can see all of these mucus secreting glands that sit in the submucosa. And these are going to have wonderful ciliated pseudostratified epithelial cells that line the inside of the mucous membrane. All right, so we have the wonderful C-shaped cartilaginous rings, trachealis muscle, and when we look at it from the front, we'll also be able to see what we call annular ligaments that go between each of those C-shaped cartilaginous rings. Annular just means circular rings. All right, so here we can see that, the annular ligaments that sit between the cartilaginous rings. So this is a nice drawing of the trachea. Again, we have the cricotracheal ligament here between the cricoid cartilage and the trachea. We have the 15 or so cartilaginous rings for the trachea. So these are those C-shaped rings, annular ligaments between them. If we look at the back edge of it, we're going to see here's that trachealis muscle. Here's the C-shaped cartilaginous ring. And then running posterior to the trachea, there's your nice esophagus. All right, so function of the esophagus is purely for conducting air further into our respiratory pathways. Especially if you've been breathing through your mouth, you're going to have a lot of particulate. So you need to make sure that what we breathe is also further cleaned as well as humidified and warmed. So we have what we refer to as a tracheal ciliary ladder. This is kind of a nice view of that. So this is that pseudostratified ciliary columnar epithelium with goblet cells in them. So these are going to be mucus secreting. Plus we also have mucus glands that can secrete a quite large volume of mucus that lines the surface of the epithelium. Here they're trying to show you particulate in that mucus, and you can see the arrow here showing you that the mucus is going to literally get pushed up against gravity up into the laryngopharynx, where it can then be swallowed. So this is a very effective way to continue to clean and remove debris that might have been inhaled. All right, so this is a histological image of that pseudostratified ciliary columnar epithelium. These lighter cells here, these are goblet cells. There's a little pore right there where the goblet cell would be releasing its secretion, and this is the cilia. So this is actually going to be moving or sort of pulsing and continually gently brushing the mucus up against gravity into that laryngopharynx so we can swallow it. All right, so this is a quick video showing you mucociliary clearance.
We breathe in a number of substances which we would prefer did not enter our lungs. Dust, dirt, soot, etc., in addition to viral particles and bacteria. These substances get caught in mucus, which lines the nasal cavity, the pharynx, and the trachea. And the cilia, which lines the upper portions of the respiratory tract, then sweep this mucus which has bacteria, dirt, etc., towards the throat. Here, it can be swallowed into the stomach. While this may not seem pleasant, bringing this dirt and microbe-ridden mucus into the stomach, which is a big bag of acid, is far preferable to bringing it to the very delicate alveoli lined by simple squamous epithelia, one of the most delicate tissues of our bodies.
Okay, so in that video again, we want to make sure that we do the cleaning of air, and this mechanism is very efficient as well as inexpensive metabolically. So we can make sure that these very simple, delicate, simple squamous epithelial cells that, remember, you have basically 2 cells away from blood, it keeps all that debris and potential pathogens out of our respiratory pathways. All right. So next we're going to look at the bronchial tree. So as we hit the trachea, and that's this midline structure, it's then going to branch and go off to the right and left of our thoracic cavities and into the surface of our lung. The bronchial tree refers to the fact that it looks like an upside down tree, with the trachea being the trunk, and then you have these branches that get to smaller and smaller and smaller divisions till eventually get to the what we call alveoli, which are like the little leaves on the ends of the twigs. Okay, so as we go further and further into the lungs, the conducting pathways are going to get more and more numerous and have more and more divisions and diversions, and also become smaller, until eventually we get down to these very teeny tiny, very small pathways through which we can then enter into these little tiny alveoli, which are going to be millimeters in diameter, and then we can have the diffusion of gases occurring. So starting off, there's a right and left primary bronchi. So this is going to be the very first branch off of the trachea. They refer to these as extrapulmonary because they're going to start outside the lung and actually go into the lung. So that's referred to as an extrapulmonary or outside the lung parenchyma. Parenchyma is a term that just simply means functional tissue. So they're outside the functional lung tissue, and then they're going to go into the lung tissue. They're going to go in at what we refer to as the medial depression, or the respiratory hilus or hilum. It is going to enter in and then start to immediately branch into what we refer to as secondary bronchi. When we look at the primary bronchi and secondary bronchi and tertiary bronchi, we're going to see they're going to be all held open with cartilaginous plates. So we're not going to see these really nice C-shaped rings anymore. We're going to see sort of a continual decrease in size of these cartilages, but we still want to make sure that the bronchi don't collapse. And so we're going to have irregular plates of cartilage that help maintain their internal space. We're going to continue to have the ciliary ladder. So when we look at the mucous membrane, we're still going to see it dominated by columnar epithelial cells with cilia. The farther we go in, we're going to see this sort of progressive decrease in height of the ciliary cells. So they're going to be going from a pseudostratified ciliary columnar down to a little bit more columnar, into a cuboidal, and then eventually to squamous as we kind of transition down further into the lung. This is looking at a slide of bronchi, and I can see here the pseudostratified ciliary columnar epithelium. These are the goblet cells, that's what the G stands for, and this would be the submucosa underneath with the basement, well, they've actually highlighted the basement membrane, that's what the BM stands for. And this is a scanning electron micrograph where you're actually looking at these little cilia coming off of the surface of those epithelial cells. All right, so these are sort of classic drawings that you might see if you Googled bronchial tree. So they typically only show the beginning of the branches. They will show all of the continual branching to simplify, and they usually only show one side. So in this case, they're showing you the left side. So this is the left primary bronchus, and then it branches 2 more times into 2 secondary bronchi, which then branch again into usually 8 or 9 tertiary bronchi, which then go down to what we call terminal bronchioles and respiratory bronchioles, and then eventually into what we call these alveolar sacs, which are these little sort of clusters of alveoli. All right, so if you were naming them in sequence, you'd say trachea, primary bronchi, secondary bronchi, tertiary bronchi, bronchioles, which are sort of the diminutive term of bronchi. We have terminal and respiratory bronchioles, and then eventually we're going to get into what we call alveolar sacs with the alveoli. All right, the only place we're actually going to get exchange or respiration occurring is down here at the alveolar sacs. Everything else is conducting. All right, so it's just another view, and they've kind of shadowed in the surface of the lung. So one important aspect is the primary bronchi are going to start outside the lung and go into the lung parenchyma. All right, so quick video going over respiratory system.
Respiratory system. To understand the process of breathing. In humans, the main organs responsible for respiration are present in the thoracic cavity. In the thorax region, the rib cage and a dome-shaped fibrous tissue, known as the diaphragm, are observed. Present within the rib cage are the pleural membranes, which enclose the lungs. The right lung is divided into 3 lobes, the right superior, right middle, and the right inferior lobe. The left lung is smaller and has only 2 lobes, the left superior and the left inferior lobe. Both the lungs are associated externally with small tubular bronchi, which unite and extend into the trachea. The trachea has incomplete C-shaped rings of cartilage, which prevent the tracheal wall from collapsing. The trachea leads into the pharynx, which is connected to the nostrils. As we breathe in air, the oxygen molecules enter the nostrils and travel downwards through the pharynx and trachea to finally reach the bronchi. From each bronchus, oxygen travels into the lungs. Within the lungs, the bronchus divides repeatedly to form bronchioles. Oxygen travels through these bronchioles and reaches the alveoli, each of which is surrounded by a network of capillaries. A section of one alveolus shows the presence of numerous alveolar chambers with pores. Blood, containing RBCs, is seen flowing through the capillaries. The oxygen molecules from the alveolus diffuse into the capillary and then get absorbed by the bluish-purple RBCs. This causes oxygenation of the RBCs, and a transition in their color from bluish-purple to red is observed. The blood moving into the alveolus contains RBCs and carbon dioxide molecules. These molecules are released into the alveolus. The carbon dioxide collects in the alveolar chamber. And then from the alveolus, it travels through the bronchioles into the bronchus, which finally reaches the trachea and is breathed out through the nostrils. So the process of breathing in air rich in oxygen is called inhalation. After the contraction of the muscular diaphragm, the lungs expand and the air rushes in, resulting in the inflation of the alveoli. During exhalation, the diaphragm moves up and the lungs contract. Thus, the alveoli deflate, causing the air to be forced out. This exhaled air is rich in carbon dioxide. This process of inhalation and exhalation is known as respiration, which is approximately 20 times per minute. Summary. In the thorax region, the rib cage and the diaphragm are observed, which play a vital role in respiration. Present within the rib cage are the pleural membranes, which enclose the lungs. The right lung consists of 3 lobes, while the left lung has only 2 lobes. Both the lungs are associated externally with bronchi, which unite and extend into the trachea. As we breathe, the oxygen molecules enter the nostrils and travel downwards through the pharynx and trachea to finally reach the bronchi. From each bronchus, oxygen travels into the lungs. Within the lungs, the bronchus divides repeatedly to form bronchioles. Oxygen travels through these bronchioles and reaches the alveoli, each of which is surrounded by a network of capillaries. As blood flows through the capillaries, the oxygen molecules from the alveolus diffuse into the capillary. This causes oxygenation of the RBCs. The carbon dioxide molecules are released into the alveolus. They are collected in the alveolar chamber, and then from the alveolus, it travels through the bronchioles. Into the bronchus, which finally reaches the trachea and is breathed out through the nostrils.
Okay, so this is looking through an endoscope that's viewing from the trachea down. So looking, this is, oh, pointing with my finger. This is anterior here, and this would be posterior. You kind of see it's a little bit flatter, so this would be where the trachealis muscle is. So if we're looking, this would be the right bronchus and the left bronchus. Looking at them, the left primary bronchus, and I made a mistake when I did the quiz, so I need to make sure I correct that, but this is a really good view of that. The right primary bronchus tends to have most aspirations in it, and this is a really good view of that. The right primary bronchus is going to be shorter and more vertical, so you can see down it really easily. The left primary bronchus is longer and more horizontal. Because the heart sits between the lungs and it's angled at about 30 degrees toward the left, it kind of pushes that left lung further away from midline. And so the trachea, as it branches into the left primary bronchus, that left primary bronchus is going to be a little bit longer and more horizontal. So more aspirations are going to occur on the right side, because the right side is going to be shorter and more vertical, which is why that right primary bronchus we can see further down through it, and the left seems to be kind of disappearing. I have there labeled the carina, which is right in the center, and that marks the beginning of those primary bronchi. Okay, so looking at this view here, we get a little bit better view of that right primary bronchus. Again, it's shorter and more vertical. The left is going to be longer and more horizontal. So that's I have here, shorter, wider, and more vertical, more prone to aspirations. The left primary bronchus, again, is going to be longer and more horizontal. You can imagine the heart would sit right here. It's going to be angled about 30 degrees toward the left, and so it's going to push that left lung out a little bit more to the left side of the thoracic cavity. All right, so once we go into the primary bronchi, we're then going to branch into the secondary bronchi, which are called lobar bronchi. And that's because, remember, the right lung has 3 lobes and the left lung has 2. So we're going to see 3 lobar or secondary bronchi on the right side and 2 lobar or secondary bronchi on the left side. We're going to find that we're still going to see those irregular plates of cartilage. We're still going to have a ciliary ladder, and we're going to see associated smooth muscle, so that in response to either an inhaled substance or an irritant, we can get bronchial constriction, which will narrow the pathways. When we want to breathe a larger volume of air, we're going to see bronchodilation. So we have larger volumes of air being moved during ventilation. So irregular plates of cartilage, smooth muscle, and a ciliary ladder continuing into the secondary bronchi. This is looking at a view of bronchi. I'm not going to require that you know the difference between primary bronchi or secondary bronchi or tertiary bronchi histologically. We want to just know that when we're looking at bronchi, either primary, secondary, tertiary, we're looking at little plates of cartilage rather than a C-shaped ring of cartilage. We're looking for smooth muscle, and we're looking for our columnar epithelial cells with cilia. So that's what I see here. Number one, this is one of those little plates of cartilage. This is hyaline cartilage. There's another one down here. There's another plate of cartilage here, another plate of cartilage there. So look for plates of cartilage. I also see smooth muscle. That's what's labeled number 2 here. So there's some smooth muscle. I see smooth muscle over here. And then I have my pseudostratified columnar epithelium. Depending on how far down you are into that bronchial tree, we're going to see that we go from a really nice, thick pseudostratified ciliary columnar to a little bit more just columnar. And as we enter into the terminal bronchioles, we're going to see that we're going to navigate in toward more cuboidal cells. All right, so this is looking at a closer up view, a much closer view. Again, really nice view of that pseudostratified ciliary columnar epithelium. I can see the end of this little cartilage plate here, so I have a cartilage plate, not a ring. I see some smooth muscle here. LP stands for lamina propria, and then down in here would be submucosa. All right, so again, ciliary ladder, smooth muscle, cartilage plates are going to indicate that we're looking at a bronchus. Okay, so next is tertiary bronchi, which we also refer to as segmental. Within each lobe, we're going to divide the lobes into segments. On the right lung, we're going to see a total of 10 segments, which are typically shown with infusion of pigment. So here we have different latex filling the different segments. So when you see these colored images, they're showing you the different lung segments within the different lobes. So there are 10 segments on the right side and usually 8 or 9 on the left. And so when we look at tertiary bronchi, they're going to deliver air into each one of those segments. So they call them segmental bronchi or tertiary bronchi. These are entirely within the lungs, along with the secondary bronchi are solely within the lungs. We're still going to see regular plates of cartilage, smooth muscle, and we're still going to see our ciliary ladder, so our columnar epithelium cells with cilia. So this is looking at, again, a tertiary bronchi. I won't have you know the difference between secondary or tertiary. Again, when we're looking, we want to look for plates of cartilage. We want to look for our ciliated epithelial cells and smooth muscle. This actually is a tertiary bronchi. One reason I can tell that is the space within the lumen is much smaller relative to the cartilage plate. But I can also see that my ciliated epithelial cells are not quite as tall. So we're starting to thin those out so they're not quite as active as we see with the ciliary ladder further up at the beginnings of the bronchial tree. But still a bronchi. So again, I'm looking at smooth muscle out here. That's the SM, smooth muscle. I'm looking at our cartilage plates, lots of cartilage plates here. I see my pseudostratified or ciliated epithelial cells here. And this is some mucus inside the bronchial space there. All right, so finally we get into our bronchioles. Now bronchioles, again, are diminutive of bronchi. So we say bronchioles to say they're smaller. We have 2 kinds of bronchioles. We have the terminal and then respiratory bronchioles. These are going into each of those segments as they branch out of the tertiary bronchi. The most notable feature as you enter into the bronchioles is the lack of cartilage. So that's what I always look for, is this lack of cartilage. Because these pathways are so small, we don't want to clog them with a lot of mucus. So we're going to see that we're going to lose the major mucus secreting glands. There will still be some goblet cells, but we aren't going to see the really big submucosal mucus glands any longer. And we're going to see that we still have some epithelial cells with cilia, but this is where we're going to start really transitioning down from a columnar down into a cuboidal. The farther you go within that terminal bronchiole. They estimate there are about 6,500 branches of terminal bronchioles off of our tertiary bronchi. All right, so just looking histologically here, this is a bronchi and this is a terminal bronchiole. And the one big difference that I can see here is this cartilage plate that sits with the bronchi. So this is a tertiary bronchi. And notice that there's no cartilage plate here. So I'm really looking at just the epithelium. I have some smooth muscle, connective tissue, and then all around, these are actually alveoli that are going to be sitting around them waiting for delivery via the respiratory bronchiole. Okay, so again, this is just another view of a terminal bronchiole. So these guys are a lot more cuboidal in shape. So they're kind of narrowing, they're still columnar, but they're narrowing down to much more cuboidal. The farther we go within a terminal bronchiole, the more cuboidal they're going to be. So you're going to start off with columnar and then eventually get to cuboidal. Notice the lack of cartilage, so there's no cartilage around here. So we just really have smooth muscle and the respiratory epithelium and the ciliary ladder there. And we don't see really any big mucus glands associated. All right, I really like this diagram because it really helped me understand the physiology, or not the physiology, the anatomy of the respiratory system. And so what I look at right here is that transition from bronchi to bronchiole. And the major difference at this location is we aren't going to see those really big mucus glands. I'm going to see that I stop the hyaline cartilage and I still have smooth muscle. I still have some elastic fibers. And notice here, this is the pseudostratified ciliary columnar epithelium. Notice it's getting shorter and shorter, more and more columnar, and then eventually going from columnar to cuboidal as we navigate down further and further into these conducting pathways. So we have one more section to go, that's the respiratory bronchioles, and then we have to be in those very thin single layers of epithelial cells as we hit those alveoli. All right, so again, this is just simply looking at a terminal bronchiole, and again, the reason I know it's one of those, terminal bronchiole is because I don't see any cartilage. So this is smooth muscle. I can see this is probably right at the beginning because I have some really nice columnar cells there with cilia. So this is one of those terminal bronchioles right at the beginning. And then I'm starting to see a lot of these little air pockets. So those are going to be some of our alveoli. This big structure right here, that is a blood vessel. So that's a big vein associated. All right, so respiratory bronchioles, these are the final pathways right before we get to our exchange surfaces. These are the thinnest little ducts that we're going to see. So for example, here we see these respiratory bronchioles coming in. When we start to see the little air sacs or alveoli associated, this is what's referred to as a respiratory bronchiole. So we're going to see that here. We still see a lot of smooth muscle, little green is elastin fibers. We get to these clusters we call alveolar sacs with each of the little alveoli chambers and alveolar ducts. So this is looking at a terminal bronchiole that's transitioning down to a respiratory bronchiole. So this is where we're going to see alveoli that are kind of coming off of it. So these are what we call alveolar ducts and alveolar sacs. All right, so definitely when we hit the respiratory bronchioles, you're going to see no more cilia. It's really nice here. You can see we go from this columnar and cuboidal cells immediately down into the cells that are going to lack that cilia. So we're going to have those cuboidal and eventually squamous cells. All right, so the respiratory zone, the only place where exchange of gases is going to occur is in the alveoli. So we call these little alveoli, little alveolar sort of sacs right here, or I should say chambers right here. These are the alveoli. You get a cluster of them. They call these alveolar sacs. And connecting into each of these clusters is going to be what we call an alveolar duct. So when you have a cluster, you're going to have a little alveolar duct that distributes air to the alveoli within that cluster. All right, so one more video, and then I think we're done with videos.
Each lung is made up of a huge network of tiny air passages. And the secret of what the lungs are for lies in the tiny air sacs at the end of each minute tube. The air sacs are called alveoli. But what are they for? Just looking at them gives you a clue. They're surrounded by a network of blood vessels. That's why we breathe, to get oxygen from the air into our blood. Let's move inside an alveolus and take a closer look at a single air sac and a single blood vessel. The air we breathe in is only 20% oxygen. The rest, nitrogen and other gases, is not much use to us, so we'll forget it. When we breathe in, there's more oxygen in the alveolus than in the blood vessel, so what happens next? It's called diffusion. Gases always move from areas of high concentration to low concentration. Oxygen molecules dissolve across the thin membrane of the alveolus into the blood vessels. This oxygenated blood returns to the heart and is pumped around the body to the tissues that need it. When you burn any fuel with oxygen, you get waste products. Blood arriving in the lungs has a lot of carbon dioxide in it, but the air inside the alveoli has hardly any. High concentration, low concentration. Carbon dioxide moves in the opposite direction from the blood to the air inside the alveoli. Breathing out removes the carbon dioxide, and the whole cycle can repeat.
Okay, so nice view of that exchange of gases that's taking place. And again, the only place where exchange of gases is going to occur is within those little alveoli and the surrounding blood vessels.
Each lung is...
All right, so now we're at the very last part of our respiratory system, and we're inside little alveoli. There's three primary cells that exist there. There's the type 1 alveolar or pneumocyte cells, type 2 alveolar or pneumocyte cells, and then the alveolar macrophages, or we sometimes call dust cells. Type 1 alveolar cells are the ones that gas exchange is occurring across. These are going to be simple squamous epithelial cells. They're going to have that very small diameter so that between the air in the alveolus and circulating blood, the distance is going to be about 1/10 of a micrometer to about 1.5 micrometers, which means the thickness of an alveolar cell is going to be less than 1/10 of a millimeter. And then the surface area, if you were to stretch out all of your alveoli, expand all those little air sacs so that they're flat, the surface area is about 1,000 square feet. So we have a very, very effective diffusion surface or exchange surface. The type 2 alveolar cells are actually cuboidal. These are referred to as type 2 pneumocytes or alveolar cells. Their job is to make a secretion called surfactant or surface active agent. This is an extremely important component that gets produced during the last trimester of pregnancy and then, of course, throughout our lives. Surfactant is a lipoprotein that helps reduce the intra-alveolar water tension. It's really important to remember that we have water lining the inside of these little alveoli, but water has an adhesion force, and water molecules like to stick together, and those can actually cause the little alveoli to collapse. So we need to have mixed with the water this lipoprotein that decreases the hydrogen bonding of water so the air sacs don't collapse. If you look at a premature infant prior to 37 weeks, they often have respiratory distress syndrome because their lungs don't make adequate surfactant, and it causes the lungs to collapse. And it makes it very difficult for breathing because they have to pull the alveoli open with every breath. These cells are also important as part of the pathology of the coronavirus or the COVID infection. The coronavirus that are part of the COVID-19 disease actually attack the type 2 alveolar cells. Their entry point is really at the angiotensin converting enzyme protein marker. And so the reason we have so many people on ventilators is because essentially it damages these surfactant producing cells, so that the lungs essentially collapse on the inside, and it gets very, very difficult to breathe. It also initiates an immune response. So we see fluid accumulating not just within the alveoli, but outside of them. And so essentially you're going to be drowning in fluid. And that's one of the reasons that we see such high morbidity with the coronavirus. So they're affecting these type 2 alveolar cells. And then we also see that, if you remember at the beginning of this lecture, I talked about the fact that the respiratory system is intricately related with blood pressure and blood volume. And it's actually through those type 2 pneumocytes. And so you see people who have hypertension that are much more at risk. There's still a lot to be learned about the coronavirus, but what we know so far is the attack on these type 2 alveolar cells. And then last but not least are the alveolar macrophages. Obviously, we want to make sure that by the time air gets to these little alveoli, it's devoid of any pathogens and any dust and materials. But just in case, we have sort of marching along the inside surface areas, immune cells called alveolar macrophages or dust cells that will engulf any debris that gets inhaled and past all of our cleaning pathways. All right, so this is just a view of the alveoli, and what they're showing you is the majority here are these little sort of, they look like fried eggs. These are the type 1 pneumocytes or alveolar cells. These are the pores or ducts that would be connecting to the alveolar ducts, bringing air in. We see cross-section through a lot of very tiny blood vessels. Those are capillaries. I also am going to see some of my surfactant secreting cells. Again, these are going to be cuboidal cells. These are my type 2 pneumocytes. And then I'm going to have dust cells or macrophages kind of cruising around the inside spaces. The type 2 pneumocytes are these cuboidal cells here. So I see these nice cuboidal cells, and then adjacent to them are these little flattened ones. These are the type 1 pneumocytes or alveolar cells. And then the cuboidal ones are the type 2. And then the guys kind of traveling across the inside surfaces, these are the macrophages. All right, and then this is the interlumen. So you see these really small pockets. These are about 1/10 of a millimeter in diameter, so nice little pockets of air. All right, and then this is another view of that. So this is one of those type 2 pneumocytes. Again, these guys are surfactant producing cells that also have the angiotensin converting enzyme on their surface. And then we see the squamous cells out here. These are the type 1 pneumocytes or alveolar cells. All right, and then the alveoli are supported by lots of elastin fibers and capillaries. So you see in these views a tremendous amount of blood flow around each one of those little alveoli. And in these drawings, what's nice is they kind of show you the transition of color from blue to red. And what they're showing you is how quickly we oxygenate that blood. So it comes in about 70% oxygenated and leaves about 99% oxygenated. I already mentioned the surface area again, very, very large surface area if you were to sort of flatten out all these little alveoli. So when you touch a lung, they feel like a sponge. So they're nice, filled with air, and you can kind of leave your little finger imprints in them. So the alveolus are these little tiny interior chambers that are required for breathing and I should say for respiration to occur. One of those side notes is damage to lungs that occurs, for example, in emphysema. If you looked at the one video clip I have of our lungs, the individual died of COPD, which is chronic obstructive lung disorder or pulmonary disorder. And in that individual, when you look and touch the surface of the lung, it's evacuated, like there's big, huge pockets inside. And that's because the alveoli have been damaged. It's kind of like taking a big sort of warehouse that would normally be filled with, millions of cubicles, and you knock down all the cubicles and then you just get left with the big warehouse. The problem with that is that you don't have the surface area for diffusion to occur. So people who have emphysema, one of the things they'll complain about is they feel like they can't get enough air in. And it's not necessarily that they're not getting enough air in, although that's part of the pathology, is that they're not able to diffuse oxygen. So they start to suffocate. All right, and there's just another view of that diffusion membrane. So you're looking at these very thin epithelial cells. So this is that type 1 alveolar cell, and this is the endothelial cell of the blood vessel. Yellow, this would be the plasma, and that's a circulating blood cell that would be carrying that oxygen away. All right, so 2 layers. We have a simple squamous epithelial cell and also a simple squamous endothelial cell that make up the diffusion or respiratory membrane. All right, and this is just a nice little scanning electron micrograph of all these little tiny air pockets, so surfaces through which we're going to be able to diffuse gases. All right, and then last but not least is just looking at the major structures of the lung. Again, we have a right lung and a left lung. They actually function separately, so they both have their own internal pleural cavity. And in fact, you can get a pneumothorax or a collapsed lung on one side, and you'll still have a functioning lung on the other. So they're in their own separate pleural spaces. In these drawings, you can see that the lung actually fills the thoracic cavity, so it goes up above the clavicles here, and then all the way down kind of into these deep recesses around the diaphragm on the right side, and then way over here toward the spleen on the left side. All right, so gross anatomy, people refer to them as sort of upside down cones. So they have a broad base and they have a narrow superior apex. They're going to be convex laterally and they're going to be concave medially. They also are going to be a little bit concave inferiorly, as we see the base is going to sit right above where the diaphragm is. And because the diaphragm has that dome shape, we're going to see that the inferior surfaces are also going to have a dome shape. Again, just referring to their orientation, we have the apex, which is superior. It goes all the way above that first rib and clavicle. We're going to see that the base is that broad inferior portion rests on the diaphragm. It's going to also be concave or having a dome shape to it. And then we have the respiratory hilus or hilum. That's going to be the entry point. It's the medial concavity where we're going to see the primary bronchi are going to enter, and we're going to see the blood vessels, both the respiratory arteries and the respiratory veins, pulmonary arteries and pulmonary veins, entering and exiting through that medial concavity we call the renal hilus or hilum. All right, so this is the renal hilus or hilum. Right, the hilum is actually a newer term. Hilus is a little bit of an older term. Most of our figures will still say hilus, but we use this term hilum as well. So in this case, we're going to see the branches. So this is the primary bronchi coming in and branching to the secondary bronchi. And then these are the pulmonary arteries and the pulmonary veins. Right, so the arteries are actually going to be blue because they're going to be coming off of the right side of the heart carrying blood needing to be oxygenated. And then the pulmonary veins are going to be red because they are going to have oxygenated blood going back to the heart to be distributed. Okay, and then the right lung, remember, has 3 lobes to it. The right lung is going to be shorter and wider, and that's because the liver is going to sit on that right side. Remember, the 3 lobes are the superior, middle, and inferior lobe. If you watch that video that I showed or uploaded of our human lung, you'll see that there's a quite dramatic depth to the lung. The inferior lobe is actually going to be more posterior. So even though we describe it as superior, middle, and inferior, it actually has a very large, we don't quite see how big it is from the anterior view, but it's a really large, more posteriorly sitting lobe. The fissures or gaps between them are the horizontal fissure and the oblique fissure on the right side. The left lung is more narrow and longer because it gets kind of pushed over to the left side of the thoracic cavity. Because the liver sits on the right side, there's a little bit more space on the left side of our body cavity. So we're going to see that the lung takes up that space on the left side. And so it's going to be longer and more narrow. The heart is going to sit with the indentation we can see right here. That's the cardiac notch, about 30 degrees twisted toward the left. So we see that medial cardiac notch. And again, we have two lobes on the left side. We have the superior and inferior. Again, the inferior looks much smaller, but it actually is quite large, and it's going to sit more posterior. So we have an oblique fissure on the left side. All right, and so last but not least is just a nice labeled diagram showing you all those different regions of the lung. So we have the apices up here above the clavicle and first rib. This is the right superior lobe, left superior lobe. Here's the renal hilum or hilus, where we have the primary bronchi or extrapulmonary bronchi coming in and then branching into the secondary bronchi. Remember, the primary bronchi on the right side is shorter and more vertical. The primary bronchi on the left side is longer and more horizontal. We have our tertiary or segmental bronchi, which are going to go out into the different segments. We'll have 10 segments on the right side, 8 or 9 on the left side. And let's see, we can see the carina here, which is the last of the tracheal cartilages and the dividing line between trachea and primary bronchi. And then we have our right medial lobe, right inferior lobe, left inferior lobe, and then the oblique fissure and horizontal fissure. Okay, we're done.